So we're just going to look at cell study and look at what the correct methodology is for drawing biological cells, biological drawing of cells, and describe how to make a temporary mount. So, in biology, you should always use single lines. It shouldn't be like sketchy lines. It should be single continuous lines. You also shouldn't do shading. Instead, we use dots. You should add labels. This one hasn't got labels, so it's not that great, but you should add labels, and your label line should not cross. And you should also add a scale bar to your picture so you know how big the specimen is. And you should always use a sharp pencil. So as an example, suggest 2 ways a student could improve the quality of her scientific drawing of this gill. So you can see already that there are lines that just stop, they're not continuous. There's shading, label lines are crossed. So all these things are things that you shouldn't do when drawing a biological drawing. So you shouldn't use shading. You should only use single lines or don't use sketching. You should ensure lines are continuous or connected. Add further labels or annotations. Don't cross label lines and add scale bars. Next thing is how to prepare a temporary mount of onion cells. So the first thing you should do is add a drop of water to the centre of a glass slide. You should then take a thin section of onion epidermis and lay it flat on the slide. Stain the specimen with iodine. Lower a cover slip gently using a mounting needle, being careful not to get air bubbles trapped. Place on a microscope slide, stage and focus on lower power first, then high.